Good morning, everyone. Welcome to the August 30th Zone Citrus Workshop. Thank you for joining us. First, I would like to thank Alison Walton from the UF-IFAS Center for her help and cooperation. Today's program offers 1 CEU for pesticide license renewal and 1 CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. Today's program will focus on late summer citrus canopy disease considerations. Our guest speaker is Dr. Megan Dewdney. She is an associate professor in plant pathology at the UF-IFAS Citrus Research and Education Center in Lake Alfred. Before Dr. Dewdney gives her presentation, I will go through some symptoms of 3 citrus diseases. Number 1 is greasy spot. Greasy spot is a serious fungal disease that must be controlled in all citrus groves. Spores germinate on the fruit and the underside of the leaves and then penetrate through the stomates. Lesions appear yellow or dark brown to black on the underside of mature citrus leaves. As the lesions develop on the underside of the leaves, they become darker and a corresponding chlorotic spot will appear on the upper leaf surface. These yellow or chlorotic spots occur when infected cells fail to develop chlorophyll. Greasy spot affected leaves fall prematurely from the tree during the fall and winter, resulting in reduced tree vigor and yield. Full coverage of the underside of leaves is necessary for best control of greasy spot, which can be achieved with higher spray volumes and slower tractor speeds. Disease number 2 is algal spot. Algal spot is caused by a green alga. More recently, the disease has become more severe in Florida and is mostly found on sweet oranges. The disease is on the branches and most visible when the alga is producing fruiting bodies from June through September. An initial symptom is bark thickening around the lesions. If conditions are favorable, the disease can kill branches of 2 inches in diameter or stunt branch growth, causing chlorotic leaves and leaf drop. The colonies vary from orange-red to dark red. They can appear as donuts with a gray center surrounded by red. During the rest of the year, the lesions fade to a much more gray-green color. Symptoms on the fruit should not be a problem because they usually develop late on overripe fruit. Lesions are dark black and circular to irregularly shaped. Disease number 3 is brown rot. Similar to greasy spot, management of brown rot fungal disease is needed on both processed and fresh market fruit. This disease is usually more severe on early maturing sweet orange cultivars. It commonly appears from mid-August through October following extended periods of high rainfall. Brown rot develops primarily on mature or nearly mature citrus fruit. The lesions on rise are brown or tan and firm and leathery. However, if later colonized by secondary decay organism, the diseased fruit soften. At high humidity, brown rat-infected fruit become covered with white mycelia. Infected fruit usually drop and have an unpleasant smell. Again, our guest speaker is Dr. Megan Dewdney. She will be talking in more details about managing algal spot, brown rot, and greasy spot. She will be talking about how to identify them and will explain the differences among them, but also where they may be overlap in management. She will also discuss what could be done for the upcoming season for these diseases. It's all yours, Dr. Dewdney.
Thank you, Mongi. So Mongi asked me to talk about citrus canopy diseases, and because we are now on the verge of September, I thought it was prudent to, rather than the early season diseases or the whole season diseases that I often talk about, to talk about some of these later season diseases that we are seeing sometimes more often than we used to. So the first one, as Mongi said, is algal spot, which is something that started to make my phone ring a lot, probably around 2014, maybe a little bit later than 2014, maybe 2016 for the first time in a concerted way. And so I started to learn more about this disease. There's not too much known. And this is something that was sort of relegated to the past. Most of the management information that we had was from the late 1960s and early 70s, so older than many of us, and is not frequent, hadn't been frequently seen, as I said, prior to 2015, and it was minor when it was seen on most cultivars, except lemons and limes. Usually this disease could be managed with routine grove maintenance in the past, and in the literature it recommended copper applications for control. And so that's what we started off with looking at. Unfortunately, this disease is likely to intensify as a problem as we get our increased average temperatures over time. We're finding this in several different crops, that algal spot is becoming more and more problematic. But it is also a concern when we have these canopies that are not as full as they used to be. So it is associated with the tree stress, and that's found on multiple hosts, and it's more frequent and severe when the trees are stressed or declining, as we see often with our HLB stressed trees. Now, I will say, though, that I have also seen this on young trees that are just coming out of IPCs, so I don't think it's just stressed trees. And so one of the speculations that was in the literature was that, you know, only healthy plants were able to form defensive structures. And one of the things that we do recommend to try and combat this disease is to reduce our plant stress as much as possible with HLB by cultural practices, you know, making sure that your nutrition and irrigation is optimized. And that's really excellent for managing our trees in general from HLB and all the other things that are out there anyway. So that's just a general disease management recommendation. And then we have disease, and then of course, then also looking at our other disease and pest management for other problems to reduce the stress. So what is this actually caused by? Well, this is actually an organism that's a little bit out of my area of expertise. I mostly have worked on fungi and bacteria over the very many years, but this is a green algae. It's known as Cephaloris virescens. In the literature, it says that it doesn't get nutrition directly from the trees, but it does cause death of the epidermal and palisade cells of leaves underneath the thallus or body of the algae, but not beyond. But that to me is a little bit contradictory. It wouldn't, if it's causing death, it seems like it could be getting some nutrients from there. Although because it is an algae, it can photosynthesize. And so, and so it's obviously generating some food of its own through chlorophyll. And how it affects bark, as it certainly does on citrus, is unclear. And this is a picture of a sporangiophore. So this is its where it produces those red-colored spores. And you can see here's the sporangiophore, here's its top, and then here are some of the spores that are going to come off, or sporangia. Don't remember the technical term off the top of my head, I'm afraid. So early infections with this disease are difficult to detect. They look a lot like those lichens that we have everywhere on our trees anyway. And they don't contain that bright red pigment, so it's not easily visible to the naked eye. I know that I struggle sometimes going, is that lichen? Is that algal spot, when it's not sporulating with its red color? It's normally also under the cuticle at those early stages, so it's really tricky to determine when infections occur, and especially since it's not visible to the naked eye. So you'd have to, look at it with a microscope or something like that. And then there are also large areas of infection that come together when multiple colonies are overlapping, or what we call coalescing. As I alluded to, it has a very broad host range. It has been observed on 287 plant species and is known to invade the stems on 80 of those, and probably more. The selection, I have a selection here of other economically important crops that are affected in the southeast. You could see here, this is blackberry canes. This has become a big problem in blackberry production. It is actually killing the second-year canes where they get the fruit. And you could see the damage of it all the way up those canes in blackberries. It is also known to cause problems with leaves on avocado, and then and in the ornamental trade, magnolia. But it also affects blueberries, and it causes cane death there, grapes, common guava, lychee nuts, mango, and also timber trees, all things that are important crops here in Florida with our incredibly varied sets of crops. So the canopy symptoms, and when I talk about the canopy, I'm also talking about not just leaves, but really the branches on the interior of the canopy. So these are the most consequential symptoms types. The lesions are usually about a ½ inch in diameter, but as I mentioned, they coalesce, and you can see things covering the whole branch as in a sheath. And it occurs mostly on branches in citrus, particularly the scaffold limbs. Most of the year, as I said before, they are this subtle gray-green color, a lot like every other lichen we have out there, and it is difficult to see, but the bark texture sometimes is different. You'll see just like a little ripple in the bark that you don't see with a regular lichen. But that's very, very subtle, especially if you're trying to scat a lot of trees. And you get that initial thickening around the lesion, not actually under the lesion, but around it. Then the lesions can crack and the bark will fall off, sort of looking a little bit like tempered glass or your windscreen when it breaks. It looks a little bit like that sometimes, and then you just get large cracks on occasion. If the conditions are favorable, we get scaffold limbs up to 2 inches being killed. So this is pretty serious, especially in small trees when you're trying to set out a beautiful canopy to hold fruit in the future years. But we also see limb stunting with chlorotic leaves and leaf drop. But most time I see mostly the cracking of the bark and then the whole limb dying back. Because I'm pretty sure that other things can get in there, some of the other fungi that are out there waiting for an opening. Lesions are most visible when there is that red fruiting, of course, which occurs between June and September. But I've had people send me pictures all the way into October, November. So I think as we get these warmer, longer seasons, we're just going to see it more and more and more. The colonies, as I said, are orange-red to dark red with a texture like velvet. And as Mongi said, they have that donut appearance where they've got a gray center surrounded by the red fruiting structures. So here's some more symptoms. You can see that fine cracking in here in between inside the areas that are fruiting. So I think the organism, the algae has grown out from the center, and now it's fruiting on the edges. And then this bit all here at the park underneath has died. Here's younger lesions. You can see they haven't got the death and the cracking yet. Ignore the red paint on here. We were trying to distinguish between our areas of where we were going to measure, but you could see that they're just coating these whole limbs here. And then here's some of the cracking that I've just seen also, rather than that fine cracking, but actually just cracking the whole bark on the limb, which is going to be catastrophic for future health of that limb. Insects and other fungi will get in that. So fruit and leaf symptoms, these are generally less severe than on tree symptoms. The fruit lesions are dark black and they're easily removed by brushing, say, in a packing house. They're circular to irregularly shaped. Under magnification, you see that they're highly branched, and they're seen on these overripe fruits, so probably off-season fruit that are still on the tree that didn't get picked. It went during regular season. There are also raised leaf lesions that occur on either side of the leaf reported, minor chlorosis around the lesions, and they will eventually apparently flake off, leaving a depression. I have never seen the leaf lesions in the grove. Perhaps I've just not been there at the right time, but since they flake off, it sounds like they are really difficult to spot because they're probably there and then gone. And if you're not there at just the right time, then that's problematic for seeing that disease symptom. So here's what the fruit diseases or fruit lesions look like sort of at a distance. And here's where you get up close. You can see that this is quite an old piece of fruit. It's kind of an alligatory skin. It's a little bit soft. And it was definitely an off-season fruit. I found this particular piece of fruit when we were picking a harvest for an experiment, and this one was not in line with all the other fruits. So you can see that branching, and this is an older picture of the algal spot on the leaves, because I still haven't seen it in the field, and so I don't have a good photo of my own, unfortunately. So that's algal spot symptoms on the leaves and fruit. So how do we control this? So I've done a few trials. I'm going to just show you one or two here. This is a trial from 2021. We were looking at Valencia trees. These were approximately 8 years old at the time of this trial. They had been somewhat stunted by HLB, and they had a lot of bare wood on the top of the canopy. That was alive, but it was bare. Applications in this grove were made with a handgun. Now that was to ensure that our wood was wetted until runoff. And we did 3 different application timings, which is a dormant spray around February 10th, a post-bloom spray on May 7th, and then when the fruiting bodies were visible here in June. We looked at a few different products. So we looked at Rivas and Arondis Ultra. These are all Phytophthora products. And the reason we were looking at these was because Phytophthora and algae are quite closely related organisms. They're more closely related than Phytophthora is to fungi because they are part of the algae group. So we had some hope that maybe these would show some promise in managing it. We also looked at Prophyte at 0.25 gallons, and then also at a gallon rate, and then Coside 3000, because copper historically has been known to manage it at 2.23 pounds. So I was looking for a fairly high rate here. And then Prophyte again at the 0.5 gallon rate, but doing that at 1 time in a year, 2 times in a year, and then all 3 applications. So I was really looking at application timing. And then we compared all of that to the untreated control. So what did we see? So we had our control here. You can see that unfortunately the Phytophthora products did not have a lot of efficacy on this, unlike what we hoped. So this is the total rating of area in a foot section of a branch, and we rated it from no bloom on the algae to and then percent coverages or percent bloom in this scale from 1 to 5. So you can see here the total rating that the Revis did the best out of the phytophthora products. It did reduce the area a little bit over the untreated control. Coside did a little bit as well, and then the prophytes, but nothing did spectacularly to really reduce the amount of total area. However, when we look at bloom, we start to see a bit better efficacy. So again, here is our control, and then we see Revis does a little bit better than the control. Coside does a little bit better than the control, but the prophytes really do a whole lot better than the control in reducing that bloom, therefore reducing the spread. So one time with prophyte at 0.5 gallons, you know, if you don't see it till the end of the season, you know, that might be, that's certainly an option. It's not bad. We get the best performance out of 3 or 2 prophyte at 0.5 of a gallon. We looked at 1 gallon. We don't get any greater efficacy at 1 gallon of prophyte, so I don't see that there's any point in doing that. And at prophyte at 0.25 of a gallon is not quite as good as the 1 or 3 times at, and this is 3 applications. And here, I'm a little bit worried that we might be underdosing here, and so I prefer that 0.5 rate where we're sort of in the middle. So, and then we did a trial again. This is the same trees, but they're a year older. The applications that were made in the same way. This time we did dormant spray, landed on the 16th of February, post bloom in May 10th, and we sprayed when the fruity bodies were visible on June 20th. Again, we looked at prophyte, looked again at that 0.25 gallon rate, 1, 2, and 3 times with 0.5 gallon of the prophyte. We use prophyte. I think any potassium phosphate would be equivalent, as long as it's rated for plant disease control and not fertilizer. We then also looked at alternate products, oxidate 5.0 with prophyte, and you can see the rate here, 0.5 volume to volume with a 0.5 gallon, and then just the oxidate by itself at 0.1 volume to volume, and that's a mix of hydrogen peroxide and peracetic acid. We then looked at Revis and Arondis Ultra, and then copper again, and then compared everything to an untreated control. Here we looked at percent area that was killed and then the percent area that was in bloom. And you can see that we didn't see a lot of kill here in the percent area that was dead. So we didn't see a lot of dead in the control. We did see an Arondis Ultra and the oxidate and Revis were about the same for that. And my technicians were looking for sort of where it had curled up a little bit and gone a little bit darker. So it didn't look as healthy as it had otherwise, but it was sort of still in that green stage a little bit, or it wasn't sporulating completely, stopped sporulating. Coside did a little bit better than the control, as did prophyte at a 0.25, but our best performing for percent area dead was the 1x, 2x, and 3x, so the prophyte, and then oxidate looked okay here, but when we looked at the percent area of bloom, we see that it's got the highest area of bloom, so there might be more dead, but everything that's not dead is blooming, which was about the same as the control. And then the prophyte is really controlling the bloom. And I think when it's mixed with the prophyte, that's probably the prophyte that's doing the most work there. Copper was sort of middling. Revis and Arondis, they did something, but they didn't do as much as I would have wanted to for putting it out, unless you had some other phytophthora problem. So recommendations based on my trial results. The phosphate products were the most consistent control over 3 years. The disease was less severe and it reduced the bloom. Much of the affected area didn't appear to be living. Pretty hard to absolutely confirm, but it sort of was flaking off and such. So we thought it was probably no longer living. The most consistent bloom reduction was with the 3 applications. So I am recommending 3 applications if you've had a severe problem in the first year. But I think in following years, you could probably just put the May and June applications out, and that would be sufficient, which would match, at least the June one would match with, say, a brown rot program. And if you've detected your problem, as I said, late in that summer, one application, I would definitely get that one application out. It will help for the next year, and hopefully you can get at least 2 applications out in the following year, but hopefully restrain it a little bit so it doesn't get significantly worse. We are starting to see it on smaller and smaller trees and smaller limbs. So that's enough on algal spot. I'm going to move over to Phytophthora brown rot. So the Phytophthora diseases basically are a group of soil-borne diseases in citrus that can travel into the canopy. As you probably are well aware, these are causal agents of root rot, foot rot, and then of course brown rot, which we're going to focus on today. We have yield reduction estimates for all of the Phytophthora diseases that were 3 to 6% per year prior to HLB, or $20 million of crop loss. Now it's a little bit harder to distinguish between what's being declined from HLB and anything else versus Phytophthora. So that's a little bit harder to estimate, but that's still a very significant chunk, I would say. So all of these diseases are connected. When we look at, the susceptibility of the roots are highest during the very wet to very dry cycles. We see wetting and drying increasing those root exudations, which is increasing the zoospores, which means that you've got further inoculum for later in the season. Phytophthora is attracted to those HLB affected roots. Some speculation is because there is greater sugar content in those roots because it's not able to move around. But together, Phytophthora and HLB are estimated to be causing approximately 27% root loss versus Phytophthora alone, which would be only causing about 10%. I would actually probably estimate a much higher root loss. I think that came from a study that Dr. Graham had done. So looking at this, the life cycle, the disease cycle here, or the life cycle, I should say, of Phytophthora, we get mycelium in the fallen fruit or infected roots. They are sometimes coming together with a opposite mating type. We get sexual reproduction, and then they go to form sporangia, and sporangia form zoospores. You can also get chlamydospores, which are those resting structures in the soil, waiting for an opportunity to form a sporangia, a signal, and then zoospores. Or the mycelia can produce the sporangia directly, and sometimes the sporangia can germinate without forming zoospores. So it's quite complicated, but these zoospores, they're called zoospores because they swim, and they could swim either to your roots or they could swim to fruit on the ground, and especially if it's wet or you're getting raining or you're getting flood or intermittent flooding. Those fruit on the ground then can become inoculum for the canopy, or you could sometimes just get soil splash up into the canopy. Then you get fruit that are infected and they go up into the canopy. So if you have Phytophthora in your grove, there is a potential for brown rot on those early season cultivars, less so on the later season ones. So, and this is because it's a coincidence of rains with the color break of the fruit. And historically anyway, most of our fruit losses are in older, larger trees. That trend seems to be still holding somewhat. And the fruit cannot be harvested until all those infected fruit fall. Neither packing houses nor processing plants want brown fruit on their lines. Hamlin is most affected, but you do see it in navel and grapefruit, and often it's showing up in those ones as post-harvest problems in the packing house. And then you get probably an angry call from the packing house. Brown rot symptoms are mostly on early season fruit. As I said, they could affect any type. We get that light brown leathery decay. You could see it here on these fruit. And we get white fungal growth on the surface under humid conditions, either under the tree or, if you're unlucky, in a packing box. Those infected fruit have a sharp, pungent odor, really doesn't smell good. You recognize it once you smell it, and you recognize it again later. And you do get infection spreading in post-harvest, and that's called nesting. It can infect one fruit's infected, it develops this fuzz on it, and then it moves over to the next fruit, and that's a process called nesting. So causal species here in Florida are Phytophthora nicotiana. It's a common cause of foot rot and root rot, but it fortunately is a minor brown rot problem. And it has these round sporangia here, if you're ever looking at them under the microscope. So that's nicotiana. And then more importantly, for the terms of brown rot, Phytophthora palmivera, which does prefer poorly drained soils, but it can sometimes be found where you do have good drainage with those high water tables. These cause sporadic outbreaks, particularly in root rot, but it can be a major brown rot concern. So this is Phytophthora palmivera sporangia. You can see they're lemon shaped, so they're pretty easy to distinguish when you're looking at them under a microscope. When we're looking at the brown rot disease cycle, as we talked about, we get the spores coming from the soil. They're affecting these fruit on the ground. They're falling for all kinds of reasons, herbicide, HLB, what have you. You get the spores on the ground, and this, if it's palmivera, you get abundant sporangia being formed, and they can splash greater than 3 feet. And then are able to actually go up into the canopy and spread throughout the whole canopy. So, you know, an easy rule of thumb is that if you've got brown rot above 3 feet, you've probably got problems with Phytophthora palmivera in terms of brown rot, whereas nicotiana cannot splash higher than 3 feet and is unable to make it into the rest of the canopy. So if you see it really restrained below approximately a 3 foot level, then you're looking at Phytophthora nicotiana. And then you can sort of see here that they're affecting roots, and they do the same thing on fruit. They come in, they do what's called encysting, so they pull their flagella in, and then they form an infection packet. You can see one here. This is the spore, and it's moving all of its cell contents into a plant intercell space where it infecting tissue, and then it's wreaking havoc. So brown rot control with cultural management, you always want to try and do that first before you have to move to the various pesticides. You know, you want to try and avoid your premature fruit drop. I realize that that's really very difficult in the HLB era, but as much as you can avoid things like canker making your fruit drop, black spot in some cases, although less common on a early season tree, but sometimes in grapefruit. And then anything like a herbicide boom knocking fruit off low skirted trees, trying to raise those tree skirts to increase your air movement, get drying of that foliage, but then also you reduce that risk of moving the organism into the canopy. So if you have a block that's been a historic problem, you should definitely be skirting that block. The herbicide strip should be just inside the drip line. And you're trying to minimize soil splash up into the canopy. And then Dr. Graham has always recommended that you put out your residual herbicides in early summer so that you're not knocking fruit off in that late summer period where we're at danger from brown rot with the herbicide boom, so that you're not coming in at that critical window. So we did a trial in 2023. This is on Hamlins and sweet oranges in a Swingle Grove. It had a history of brown rot. It was quite wet in that grove. We're worried about getting stuck. The treatments were applied with a handgun. We put out one application of our products on October 9th, 2023. And then we collected fruit from the trees on day 1, 15, and 30. And then the fruit were inoculated in our lab with Phytophthora palmivera zoospores. And that was just to ensure that we had enough disease to make sure that we could see a result. We then incubated those fruit for 15 days and rated them for disease. And we also included an uninoculated control to see the natural disease pressure in the grove. And that was just to see what we would have if we hadn't put our inoculum on there. And it does give you sort of an interesting idea of what you might see. So we looked at Revis and Arontis Ultra. Those are the two newer products that are recommended for brown rot control. And then we compared that to Prophyte at 4 pints per acre. These two were applied at 8 fluid ounces per acre, and everything was compared to an untreated control. So a fairly simple trial. And then we looked at the presence or absence of brown rot. Was the fruit infected? Yes or no? So looking at our two controls, we had between 60 to 100% of the fruit being infected, depending on the time frame that we looked at the fruit. I'm not sure why day 15 we found so many more fruit that were infected. And you can see that's fairly consistent among all the treatments. Not quite sure what happened there, but that was the data we had. And then we had, here's our uninoculated control. So this is our untreated control. And you can see that the basal rate of brown rot in this grove was about 20%, which is pretty high. So everything worked better than the untreated control for our products. So that was Orondis Ultra. Sorry, I forgot to put the Ultra on there. Prophyte and Revis. And then you see that all of the dates. Now, interestingly, Prophyte seemed to get better over time. And that is sort of consistent with how Prophyte works. It does have some direct effect against Phytophthora, but it tends to increase the stimulation of the plant defenses over time. And so that is consistent with what we might expect. We see our best results with the Orondis and the Revis at day 1, which is not too surprising, right after treatment, but they held up pretty well, and everybody was consistently better than the control. And you can see sort of break the potentially starting to lose efficacy by day 30, although Revis is doing fairly well by day 30 still. So that's what we saw in terms of presence or absence. Was the fruit infected, yes or no? And that was after we put a big glob of the fungus, or not the fungus, the omycete on top, the pathogen. And then when we looked at the percent surface area infected, again, here is our uninoculated control. We see the little bit, 20% and under approximately, and here are our control, our inoculated control. We see the least surface area being infected on that day 1, which was sort of interesting. Maybe the fruit were a little bit less susceptible, but it picked up on day 15 and day 30. And then we see that all of the other treatments are significantly better, except for Prophyte on the first day in terms of restricting lesion size. Now, of course, the real interesting point with, and this is sort of be looking more at sort of your inoculum control for future outbreaks, of course, because once a fruit is infected in terms of brown rot, it's no longer going to be useful for harvest, right? Nobody's going to want to process or pack that. So looking at fungicidal management of brown rot, I should say, not brown spot, is often the single spray only. If you know that you've had a lot of problems late July prior to disease symptoms, you could put that out with one of your potential algal spot sprays. And we would recommend the products for that time would be Alliet, Fostrol, Prophyte. These are all PO7 things where they're doing that defense stimulation, and they're estimated to have 60 to 90 days of control after application. If you find symptoms and you didn't put out your Prophyte, your phosphite kind of products earlier in the season, but you find you're having a problem, there is the option of putting out copper. In August, if you're going to use copper, it would be sort of that August timing, which is primarily a protective, but it can kill inoculum if you're seeing it. And there's only about 45 to 60 days of control with that. If you need an October spray and you're finding brown rot where you didn't think you were going to have any, you of course could use copper again. I would certainly look at that Revis and Orondis Ultra as good choices for the late season if you missed that earlier window. It worked really well for us. I think it held the disease to about native levels. So you're going to get at least 30 days protection. That's as far as we tested, so I can't really say about further. And we did add those to the recommended product in the production guide in 2024. So that gives us new options. I'm glad to have some products to rotate. There are reports of things like phosphite resistance in California, where they've used them pretty heavily for a long time. So I'm really hoping that we could avoid that by having some rotational products in the set of things that we can use that have good efficacy. So I would certainly recommend Rivas or Arontis Ultra, and you may be able to use them earlier in the season. I just don't know if they'll have that same, if they'll have as long a efficacy window as the copper or the phosphites. Okay, so final topic, greasy spot. So looking at, so as Mongi said, we get a lot of these funny looking lesions on the leaf surface. We get a bit of chlorosis around them. You can see that particularly here. That chlorosis fortunately greens back up if the leaf doesn't drop from the amount of disease that it would be on leaves like this. Grapefruit, you see a lot of sort of cloudy lesions. Sometimes it's kind of indistinct. You could see when you get up close, though, it's a lot of pinpoint lesions, and then they start to coalesce. You get some pink pitting. And then this is if you're trying to degreen fruit, this area is likely not to degreen. And then you see here also the effects. You know, here's some degreened fruit, and they're trying to pack that, and it's just absolutely covered with black spots, which is not acceptable in most markets. And then you see also these are leaves that are pretty badly affected, and that one's probably going to upsize a little bit earlier than it might otherwise without all that infection. So the disease cycle, this organism recently had a name change. It says Midium citrogricium now instead of Mycosphaerella citri. But it's the same organism. Just as a refresher on the disease cycle, remember we get leaf drops for the February, March. Those leaves start to decay. You get these structures called pseudothecia forming in the decayed leaf litter. The ascospores eventually mature. They start to eject. They start to be ejected with wetting events. Now, the unique thing with greasy spot is then we get on the underside of the leaves in particular, and also on the fruit, you get what's called this epiphytic, or on the surface of the plant growth. Sometimes you might find some very minor number of conidia. Then by the end of the late summer, we start to get infection through the stomates. On warm years, we will see lots of fruit and leaf symptoms in November, December. I often wait till January to rate greasy spot, because then I'm sure that all the disease is out there, because it can develop through that winter depending on the ambient temperatures, and then the whole cycle occurs again. So this is really what we would consider a monocyclical disease. So there's only one major cycle through the year, and we don't really understand the role of the conidia, if there is much of a role in this particular disease at all. So inoculum production, just as a reminder, our primary inoculum is coming from our leaf litter. Those ascospores are ejected during wetting events, form structures in partially decomposed leaf litter. And the wetting and drying of litter is really important for spore formation. And you can see that historically, way back when Jack Whiteside was still alive and practicing plant pathology in Florida, our major ascospore ejections were actually occurring in June and July. As new irrigation methods came through and we were favoring the disease with our wetting and drying on the leaf litter, we actually shifted the majority of ascospores being produced into May and early June. And while that's actually been quite beneficial, because the majority of the ascospores are missing the susceptible flush. However, it doesn't take a whole lot of spores to cause disease, because of that epiphytic growth stage, where it only takes a few spores to then proliferate all over the leaves or fruit. So I did a rind blotch trial in 2022. This was on white grapefruit in the Indian River region. We applied treatments on June 7th, 28th, July 19th, and August 9th. And they were all done at handgun again with a 200 PSI. And then we collected data in January 2023. And we looked at a rating scale of no lesions up, and then we looked at the percent surface area on the rind blotch. So from 1 to 4%, and then all the way up to greater than 50% of the tree, or the fruit surface area, excuse me. We looked at quite a few treatments. So we looked at double nickel and with its cohere as a surfactant, double nickel and cohere alternated with Coside 3000O, double nickel and cohere with Coside 3000O all at the same time. So we're looking at a quart, 0.125% volume to volume of the non-ionic surfactant, and then 1.5 pounds of copper. Then we looked at copper hydroxide or Coside O by itself, and we did 2 applications at 3 pounds and then 2 applications at 1 pound. We then also looked at enable alternated with Coside 3000, where that 8 fluid ounces and 1 pound of copper. And we looked at another new product called Aura Citrus. I don't know if they've got greasy spot on the label, but we did try it. 1, 2, or 3 applications with their proprietary surfactant K122, and at 34 ounces per acre, and then oxidate with cohere, oxidate with 435 oil, oxidate and copper, looking at different levels or quantities of oxidate. So at 1% and 5%, 3% oil, 1.25% of the cohere. And then the oxidate here was put out at 0.5 volume to volume. And then the copper was put out at 2 applications of 1.5 pounds and then 2 applications of 0.75 pounds. Little slightly more complicated treatments than sometimes we see. So, and then we also looked at straight 435 oil at 4%. And then I looked at Moravis Top, which is one of the newer fungicides on the market because I didn't know much about it. And then an untreated control. So what did we see? So this is looking at our rind blotch disease ratings. Everything performed better than the untreated control. Here is our 435 oil. It is usually the worst performer in terms of control treatments for rind blotch. It does decently well on greasy spot on the leaves, but on rind blotch it seems to be less durable. We looked at oxidate and all the Aura citruses. They don't do particularly well, but we do get excellent control from enable alternated with Coside, and then the double nickel cohere and Coside treatment. Also pretty good control with the alternate with Coside. So if you wanted to save money or you just want to reduce the amount of copper, that looks like it's a pretty good alternate. This Moravis Top is looking very good in this first year of trialing, and Coside 3000 was pretty reasonable again. The double nickel with the cohere itself wasn't too bad, but it looked like that copper gave it a little extra kick. When we looked at the number of marketable fruit, so this is anything with a rating of 1 or 0, so less than 4% of the surface area being affected. As you would expect, the untreated control had only about 15% of the fruit being marketable. The oil was 25%, so not terribly amazing. Our best treatment, the double nickel cohere and Coside, had about 60% of the fruit that were marketable. Enable was sort of around 55%, and then several of these others like Moravis Top, the alternate, and the straight cohere, and then Coside 3000 were all equivalent in terms of marketability. And then you see that there is an effect of some of these things like the Aura Citrus and the oxidate. It's just not as good as some of these other ones. So they do have an effect, but I don't think it's enough to consider using them on your fruit regularly. So looking at control products, coppers of all formulation, strobilurin and fungicides, the Abound Gem and Headline Amistar Top contains a strobilurin, as does Pristine. These are all to be used only once for greasy spot, anything containing a strobilurin. And there are concerns out there about strobilurin resistance, and it's something that we are actively trying to investigate. It's just very difficult with this particular organism because of its nature and how easy it is to work with in the lab. So bear with me. We are actively exploring this subject, and I hope we'll have an answer in about a year. Further products, fungicide efficacy, fungicides that are effective for, further fungicides that are effective for greasy spot enable. It's still holding up. It's a good substitute for copper in those hot times. If you're really concerned about stippling, I would and you're don't have too many concerns about canker, I would certainly consider it, because it's still working well. Double nickel looked pretty good, and Moravis top, but at the moment I only have the 1 year of data. So I do want to put that caveat out there, but I thought they looked very promising. Petroleum oil, as always, less consistent for control on fruit than copper, and it can be mixed with all the other, fungicides for a little extra oomph sometimes. I do want to mention that we had every intention of rating leaf data in this trial. However, there were very, very few greasy spot symptoms on the leaves, which was a bit of a curiosity, and I don't have a fantastic explanation as to why that might be. I do want to acknowledge all the people that contributed to all of these trials: Tracy Hobbs, Etelvina Aguilar, Monty Myers, who works down at IREC, and then Ava Munlandeza, who is my graduate student who works a little bit with Monty, but then also is working on that fungicide resistance question. And I will be happy to take any questions if we have time. Do we have time, Monty?
We have time.
Monty?
Thank you, Dr. Dewdney. Any question or comment? Again, if you need CEUs, email me your name, email address, and license number. Looks like no questions, no comments.
All righty.
Thank you for joining us, and see you next month.